Today we'll talk about this gene that we hadn't seen before called PCSK9 and its relationship to a gene that we had seen before called LRP1. And the paper that we'll discuss today briefly is PCSK9 acts as a key regulator of A beta clearance across the blood-brain barrier. Cool, so in the introduction, the authors introduce for us, and in the context of this class in a way remind us of the A beta cascade hypothesis that says that accumulation of A beta and eventually the formation of plaques that are toxic to neurons is really a result of an imbalance between production and clearance of cerebral A beta oligos. And what they mean by that is the production of A beta peptides is a normal process, even in healthy individuals, and that those A beta peptides are normally clear through a series of active processes from the brain, and therefore they do not accumulate and form the toxic plaques. But when something fails in this mechanism, there is this imbalance between production and clearance, maybe same levels of production but reduced clearance, or in some cases there may be increased production and reduced clearance, which obviously leads to the accumulation of toxic levels of this thing. In fact, previous research showed with 80 patients showed that production is often not the problem. That what the problem is the lack of clearance of these A beta peptides from the brain. So it's not like this. We've seen a number of papers and a number of mechanisms by which the production of A beta peptides is increased due to mutations, due to changes in gene expressions of various kinds. But when you look at patients, in many of them, production is not the problem. It's the lack of proper clearance that is causing the accumulation of A beta peptides. All right, so one of the main regulators of A beta clearance from the brain is this membrane receptor called LRP1. It's a receptor, one of these receptors that it's a receptor, but it's actually a transporter. So it binds to A beta on the cerebral spinal fluid side of things, so the liquid inside of the brain, and it takes A beta from there and moves it through transcytosis into the bloodstream such that blood can take it away from the brain, right? It turns out that LRP1 is degraded by this protein that we're concerned with today, PCSK9, and previous work showed in other tissues, particularly in liver, that PCSK9 can regulate LRP1's close cousin, the LDL receptor. And if LDL kind of rings a bell, these are things that doctors measure when you go for blood work in a regular visit, because LDL cholesterol levels are an indication of how low or high or normal your bad cholesterol levels are. So that's where LDL may ring a bell from. LDL receptor is the protein that on the surface of cells in the liver will bind LDL cholesterol and then move it into into cells, take it away from the blood, and therefore reducing cholesterolemia. So the way this works is the LDL receptor will bind the complex between LDL, which is just a transport protein, and cholesterol, and bring it into cells through endocytosis, right? And in early endosomes, the LDL receptor will separate from LDL cholesterol. LDL cholesterol will go its way and be destroyed in lysosomes. LDL receptor will go its separate way and be recycled back to the cell surface so that it's ready to catch and bring in the next LDL cholesterol complex, right? So that's what happens normally. But if PCSK9 is present in high levels, it will bind LDL receptor and doesn't let it to be recycled. It sort of forces it into the lysosomal pathway, and now the LDL receptor is not recycled, it's destroyed. So there won't be any more LDL receptor to catch the next molecule with cholesterol on the surface. So if PCSK9 is present, LDL receptor is chewed up instead in lysosomes. Previous work showed that if you block PCSK9 with antibodies that bind PCSK9 and don't let it do its normal jobs, then the surface LDL receptor goes up. So it's an increase in levels of LDL receptor, and cholesterolemia, meaning the levels of cholesterol in blood, go down. So essentially, and I copied this straight from the paper, what the authors hypothesized is that the inhibition of PCSK9 by these FDA-approved monoclonal antibodies might, just like they help with LDL receptor in the liver, they might help with keeping higher levels of LRP1-mediated A-beta clearance, and thus be a potential therapeutic approach to Alzheimer's disease. So these drugs are used to reduce levels of cholesterol by acting on LDL receptors in the liver. What they're saying is maybe we can use these same drugs to reduce the levels of A-beta in the brain and therefore help with Alzheimer's disease. Okay? So that's the proposal, that's the hypothesis, and that's what they want to study in this paper. So the first experiment they do is they take these B end.3 cell line. These are cells that were obtained from brain, hence the B, endothelial cells, hence the end. So this is a cell line that was established in vitro through immortalization and the usual from brain endothelial cells. And what they do is they take the parental cell line, in this case indicated with this white bar, and then they generate stable clones that will overexpress PCSK9. So the approach they're taking in this experiment is, instead of taking out PCSK9 to see what it does in these cells, what we'll do is we'll put more of it in them, right? And with these cells, what they do is a transcytosis assay. So they plate the cells in a monolayer over a surface that is porous, and then they essentially load the top of these cells with A-beta peptides that are radiolabeled. So these A-beta peptides are radioactive. And then they let these things be moved by transcytosis to the bottom compartment of these special chambers, and all they do is measure the difference in radioactivity after a little while. So the more transcytosis there is, the more radioactivity they will detect at the bottom. And what they plotted in this graph is the normalized transported. So this is measurements of how much of this A-beta was transported from one chamber to the other in cells that were, in a way, normal, though they are immortalized and living in culture, et cetera, or cells that express a lot of PCSK9. So in the PCSK9 overexpressing clones, they see a reduction, significant reduction in the transport of A-beta, right? They tell us in results that this reduction is about 42% in the transport of A-beta. This is an in vitro assay, so they're looking at cells in vitro. But essentially what they're doing is they're supporting this hypothesis, and remember, this is the hypothesis they start with, that PCSK9 would normally inhibit the activity of LRP1, and LRP1 normally induces A-beta transport. So PCSK9 inhibits LRP1, which means that indirectly PCSK9 would be inhibiting A-beta transport, right? It makes sense. If a molecule inhibits an inducer of a process, then that molecule will be indirectly inhibiting the process, right? When they put in a lot of PCSK9 through overexpression in these cells, theoretically what's happening is there is an increase in the inhibition of LRP1, and therefore there is this much reduced A-beta transport. So what these data are showing is they're showing the connection between PCSK9 overexpression and the final outcome, which in this case is reduced A-beta transport. We've been here a number of times before, so I'm asking you, how could they further support this model? What experiments could they do to further support this model? Knock out the middleman, right? So what would that look like? Well, how would you design the experiment? So you're inhibiting LRP1, you told me, by CRISPR or some other means. You're measuring A beta transport. What else did you do? Do you do anything to PCSK9? No? Uh-huh. Hmm? Decrease PCSK9? And then inhibit LRP1 as well? Or not touch it or do anything to it? I know Jessica was... Okay, so we're talking slightly different experiments. I'll address that in a second. Let's stick with Gabby's experiment. So, Gabby, towards the end, you said, no, maybe I would overexpress PCSK9. Did I hear that correctly? Yeah? And that's right. In these famous, quote-unquote, take out the middleman experiments, what we want to do is we have the original trigger, if you will. We do have the overexpression of PCSK9, and then we do look at the outcome. What we're doing in these experiments is taking out the middleman separately, right? So what we're doing is we're inhibiting LRP1, and essentially the question we're asking is, do I still see the effect of the original trigger if I'm taking out the middleman? If I'm taking out that intermediate step, or the intermediate step that I'm proposing, when I take out that intermediate, I should break the link between PCSK9 and A beta transport. I should see that now overexpressing PCSK9, which we're doing here, has no effect on A beta transport, right? Now, there were other experiments proposed. So Jessica said, you were saying increase PCSK9 and then also experimentally increase LRP1. What would you expect to see here? What would be your prediction that supports this model? Go up and one or both. And would there be a difference between the both and the one? Would you expect to? Yes, right? So what you're saying is PCSK9 plus more LRP1, we might perhaps enhance even further the, rescue the, So what you're proposing is a rescue experiment. So the idea here is if PCSK9 is indeed inhibiting LRP1 through whatever mechanism, if I put back LRP1 via some independent experimental approach, through overexpression, through whatever, injecting mRNA, whichever way we can choose to put back LRP1 in those cells, then I should see that now A beta transport is restored. If A beta transport was being regulated by PCSK9 through some other LRP1 independent mechanism, then adding LRP1 or not should make no difference. But if I see that A beta transport is restored, then we've rescued A beta transport, and therefore that would support the model. Cool. And anything else?
How about we try
Inhibiting
inhibiting the PCSK9?
PCSK9. So if the model is true, what would you expect to see with A beta transport?
If it gets inhibited, then there would be more LRP1.
Uh-huh.
And then therefore more.
Okay, cool. See, all of these ideas are valid, and they're all complementary approaches. So Melita's idea is, instead of increasing PCSK9, what we do is inhibit it. So now we have PCSK9, but now we inhibit it somehow. And what we expect to see is a lot of LRP1, and therefore a lot of A beta transport. I see. So what you're saying is having LRP1 present or not? No, sorry, you said LRP1 present and then PCSK9 present or not?
Yeah.
Is that what you said?
Yeah, and then another one that has no LRP.
Okay.
And then PCSK9.
Okay. So what you're addressing in a way is one of these sort of epistasis experiments, right? If the pathway is PCSK9 LRP1 effect, then you would expect that when you manipulate PCSK9 one way or the other, and LRP is there, then you should see the effect going one way or the other. But if you take out LRP1 now, the effect should go away because LRP1 is mediating the effect of PCSK9. Whereas if it were the other way around, you would manipulate LRP1 and see the effects on A beta transport. But if you take out PCSK9, maybe it was the other way around, right? So the experiment that you're proposing, in a way, is a combination of both. So it's a combination of this experiment here and a combination of this experiment here, right? Cool. Any ideas about, so when they did this experiment, they went for, yeah, Risha. Mm-hmm. So the idea here is we increase PCSK9 as we did with these cells, but we also simultaneously inhibit LRP1. And the question we're asking is, do we still see that A beta transport reduction that is normally caused by having more PCSK9? Because if we do, then it means that LRP1 had nothing to do with this, and PCSK9 was acting through something else. But if the ability of PCSK9 to inhibit A beta transport depended on LRP1, then those experiments in which we take out separately LRP1 should show that now PCSK9 can't do anything. Yeah. So the point here is that doing all of these experiments obviously would lead us a long way, but we also need to sort of address this link here, right? The proposed model is that PCSK9 is acting through LRP1 in the sense that it is controlling its levels. So in order to fully support this model, what the authors should do, and they did do, is take a look at this connection here and really say, okay, is it true that when we put in a lot of PCSK9 in cells, LRP1 is inhibited? Sometimes when we say that a certain regulatory connection depends on a given molecule, so when we say all of these experiments up here that you guys proposed address the dependence of the effect that PCSK9 has on A beta transport on LRP1, right? We're constantly asking, is this intermediate needed or not? But what the model says is a little stronger. What the model says is not only is the effect of PCSK9 dependent on LRP1, but also we're saying that PCSK9 inhibits LRP1. And they should be able to show that, right? And they did. In the one of the next experiments they do, they take these cells, the BN3 cells, control or those expressing high levels of PCSK9, and they look at LRP1, the alpha chain. There is a beta chain that they probe for later. And as you can see in these plots, there is a noticeable reduction in the amount of LRP1 in these cells when they overexpress PCSK9. So in other words, this part of the model, this part of the hypothetical pathway is supported by the data. They put in more PCSK9 in these cells, they see less LRP1. Okay? And then of all the experiments that you guys proposed, the one that they show us next is the one in which they take out that intermediate, right? So what they did is they took these cells that overexpress PCSK9, shown here, but in addition to having the cells overexpress PCSK9 or not, what they did is they added a blocking antibody against LRP1, right? So they're not taking out LRP1 via CRISPR or RNAi or some pharmacological inhibitor. What they're doing is they're adding an antibody that will bind LRP1 on the outside surface, obviously, and block LRP1 from doing its normal job. So for all intents and purposes, this is like inhibiting LRP1. And when they did that, what they saw is that now adding a lot of PCSK9 didn't really change AB transport. When you compare these two bars, there's no significant difference between them, right? with taking away the ability of PCSK9 to do anything to A beta transport. Now you may notice that both bars are low. Why is that? So there's no difference between them, but then also they're overall lower than control cells. Why is that? That's right. That's right, because we don't have LRP1, and that goes with the model. If you take away LRP1 from these cells will have a reduced ability to move A beta through transcytosis. So it's no surprise that taking away LRP1 from these cells will produce these significant reduction from control cells. And what this experiment is telling us is if you take those cells without LRP1 and you now overexpress PCSK9 in them, you won't see any effect from PCSK9 overexpression. And the conclusion from that is PCSK9 needed LRP1 in those cells to do anything. It's not acting through anything else. One last question while we're at it. So the ability of these cells to move A beta via transcytosis is reduced to about 50%, but not completely abolished. Why do you think that is? Yeah. Some secondary process, you call it, some independent mechanism that acts along with LRP1 to move A beta across these cells, and that secondary process is not being affected by PCSK9. Cool. Any questions, anyone? Comments? Ideas, thoughts?
What is the plus IgG again?
The one?
Plus IgG.
Plus IgG. Oh, excellent question. So whenever, that didn't show up in the previous graph because I had blocked it. Whenever you treat cells with blocking antibodies, like in this case, you incubated the cells with an antibody that blocks LRP1, there's always the lingering question in the back of our minds. Was it blocking LRP1 or was it bathing cells in antibodies? Maybe these cells don't like to be swimming around antibodies. Who knows? Maybe these cells react to the presence of antibodies in general, regardless of what they target, right? So as a control, what you do is you incubate the cells with what is called a generic IgG. And by generic IgG, it's essentially an antibody that doesn't recognize anything. Even though that's a little crazy because all antibodies will recognize something. These IgGs are, I don't remember details in this experiment, but you can make them recombinant such that they don't express the portion of the antibody that recognizes an antigen. So essentially what you're doing is you're incubating the cells with an antibody that looks like an antibody, but it's not an antibody. And it's a control for just seeing how much of what you see with your experimental antibodies may be real versus an artifact of having these cells swim around antibodies all day. Yeah? All right, so let's keep going. The question they asked next is, all right, we saw all of these with this cell line, the bend.3 cell line. Can we see all of these with other mouse cells? And they repeated the experiments with these BMEC cells, and that stands for, I don't know why they flipped the words, but it's like that in the paper. BMEC stands for brain, mouse, endothelial cells. I would have called it mouse, brain, endothelial cells, but brain, mouse, endothelial cells. And in a nutshell, like I could show you this graph and the previous one, they're almost identical, right? So same idea. In wild-type cells that you're not blocking or that have normal levels of LRP1, there is a drop in A beta transport when you overexpress, or when, in this case, you're not overexpressing PCSK9 from the inside. They added PCSK9 from the outside. So they express PCSK9 in vitro, in like bacteria or whatever. They made recombinant PCSK9, purified it, and then they added it to the cells in the culture media. And when they did that, there is this drop in A beta transport. On the other hand, if they take these BMEC cells that lack LRP1, these were obtained from mice that are knockouts for LRP1. Now, the effect of adding PCSK9 does nothing, or is zero. Okay? And overall, there is also that significant drop in the transport of A beta, but not full blockage, because as Lucy was suggesting, there may be additional mechanisms that don't depend on LRP1, but are there to transport A beta across the cells, and those mechanisms are PCSK9 insensitive, if you will. Right? They won't respond to levels of PCSK9. From the previous experiment, the changes in the previous experiment, PCSK9 was being expressed by the cells themselves. Right? So we're increasing the expression of PCSK9 in the endothelial cells themselves. Here, PCSK9 was added from the outside. It was added with a pipetter to the culture medium. It was produced somewhere else and added to the medium. Still in vitro, yeah. Still in vitro, still one of these transcytoses, or the experiment itself is the same. So you have this porous membrane, you plate the cells on that porous membrane, you add the radioactive A beta to one side, and then after a little while, you measure how much radioactivity made it through. Yeah. Okay. So, yes, they can see with other mouse cell lines. They also tried with porcine brain endothelial cells. So these are endothelial cells that were isolated from brains from pigs. And in this case, they don't have the luxury of isolating brain cells from, or brain endothelial cells from LRP1 knockout pigs, because the knockout technology in pigs is not as, it exists, but it's not as easy as in, you can make a knockout pig, I suppose, if you want, but it's way more expensive and laborious than making a knockout mouse. So in this case, they don't have LRP1 knockout cells to test that aspect of the model. But what they do have is these porcine brain endothelial cells, PBECs, in vitro on these porous membranes, and they add radioactive A beta to one side. They measure radioactivity on the other side after a little while. And what they see is that when they incubate these cells with recombinant PCSK9, there is a significant drop in A beta transport. So less A beta is making it through these cells when these cells are exposed to PCSK9. So the model was supported in two different cell lines from mice and one cell line from pigs. I mentioned that they don't have access to the LRP1 knockout porcine brain endothelial cells. What else, what other approach do you think they could have used to test for the requirement or the dependency of PCSK9 on LRP1? If you were doing these experiments and you said, well, I can't make a knockout pig for LRP1, that would be way too much work and money and time. What else could you do to see if what we see in this graph here depends on LRP1 or not? Inhibit PCSK9, you said? That's it. That's it, right? So that's without PCSK9. This is with. But the question we're asking is about the dependence on LRP1.
Could you just inhibit the LRP1?
Cool, and how would you do that? Exactly, the same antibodies that they used in the previous experiment. Was that your idea too? Yeah, that's right. So there may be a number of ways in which you can inhibit LRP1 expression in these pig cells. You could probably try CRISPR. You could probably try some sort of RNAi, right? You could probably try some pharmacological inhibitor. All of those are valid approaches. But it turns out that they do have a LRP1 blocking antibody. They used it in the previous experiment, right? So they could take that antibody and block LRP1 in the pig cells. Absolutely. And to be honest with you, I think that these blocking antibodies would probably achieve that. Yeah. Instead of total ablation, it's probably a reduction.
Yeah, like for
Yeah.
For
Yeah. And even if you don't get, here's the key. So with blocking antibodies, I suspect that you're not going to get rid of every functional LRP1 molecule, and yet you still see, at least in the previous experiment, right? You still see the loss of effect of PCSK9. So that's pretty strong, because basically what you're saying is, even if we don't get rid of every single LRP1 molecule like you would say in a knockout, I pointed it to the knockout, but the experiment before, okay? Even if you don't lose every single LRP1 cell, you might still see an effect. I want to make sure that I give everybody a chance to speak. So when I asked that question before, there were other hands up. So you said that you were going to propose the antibodies, same thing, yeah? Yeah. Whenever you're treating with antibodies, and this goes from experimental animals in labs to people, when you give them therapeutic antibodies, it's all injected in blood. Yeah, so antibodies are not designed. What you do is you take an animal that can make antibodies and you inject them with a lot of LRP1, and that animal will start making antibodies. So it's not that no one designs those antibodies. The immune system will make antibodies against all kinds of molecules. And when you inject an antigen into an animal that can make antibodies, the antibodies that recognize that antigen will start being produced in large amounts. That's the essence of vaccines. When you vaccinate someone, what you do is you inject the antigen that you're hoping the immune system will recognize later. And what you're doing is you're boosting the production of cells that can make antibodies against that thing. In this context, what you would do is you would probably take an animal that is not related to pigs or mice. So you can take, say, a chicken, right? And then you inject LRP1 into those chickens. Those chickens will make a lot of antibodies against LRP1 because the amounts that you're injecting are like just insane. And then you take those antibodies from the chicken, and those antibodies now are ready to recognize and bind and block LRP1. Notice, I didn't want to go there, but I'll address it briefly. In your idea of using the LRP1 antibodies that worked with mouse LRP1, in this experiment with pig cells, what assumption are we making? Jacob? That they're not related? That they're related, right? So we're making a huge assumption here. We would make a huge assumption. That is that the antibody that recognizes and binds effectively to mouse LRP1 will also work to bind and block pig LRP1. And that may be the case, may not, it depends. It depends on how evolutionarily conserved these things are, and in this case, how distant from each other mouse and pig are. There's a high chance that they may recognize each other. And then I mentioned before, you could use a chicken. Well, a chicken is further out, evolutionarily speaking, so there's a good chance that when you inject a mouse LRP1 into a chicken, the chicken's immune system says, what is this? Because its own LRP1 is different enough that it will recognize it as something foreign and weird and potentially harmful.
When you're injecting the antibody, would you inject in the control like a saline as a control to simulate, or is that?
Okay, so that's an excellent question. So when you're doing this, you're doing it for, it's almost like for antibody production purposes. So it's not an experiment. You're just trying to get a tool, right? You're trying to build a new tool. So no, chances are that you would take chickens, and I keep saying chickens because that's probably the approach that one might take, I don't know. And then inject chickens only with LRP1, not saline. You would do the saline if you wanted to sort of find some sort of difference between the chicken that did produce the antibodies and those, but those are research questions that don't apply here. All you want is an animal to produce antibodies so that you can take to the lab and use them in a research context. Yeah. Any other questions, anyone? Cool. So next, they take the complementary approach. Instead of overexpressing PCSK9 in these cells or bathing them in recombinant PCSK9, in other words, increasing the levels of PCSK9 in these cells, what they do is the opposite. They incubate the cells with a PCSK9 inhibitor. And in this case, they don't incubate cells in vitro. They're exposing cells in vivo to the PCSK9 inhibitor. And this PCSK9 inhibitor, it's actually a blocking antibody, one of these famous FDA-approved monoclonal antibodies against PCSK9. So they're using things that could potentially be used with humans in therapy because they've been approved by FDA already, meaning we've shown that they're safe, et cetera, et cetera, et cetera. So they're taking these antibodies that exist already and injecting them into mice. And the question they're asking now is, all right, we know that elevating the levels of PCSK9 leads to reduced A-beta transport through transcytosis. If we now block PCSK9 activity, what would you expect? Yeah. The opposite, increased transcytosis, increased clearance, right? So you would expect that now these animals get better at taking A-beta out of the brain. Because according to the model, less PCSK9, more LRP1, more transcytosis, less A-beta left in the brain. So what they did is they measured the picograms of either soluble or insoluble A-beta 42 left in the CSF, in the cerebral spinal fluid, of mice that were either control mice or mice where they had injected the PCSK9 inhibitor. So for both readouts, the soluble A-beta present in the brain and the insoluble A-beta present in the brain, what they see is that there is less A-beta left behind. I honestly wish they had displayed this slightly differently from the clearance point of view, so that they show that there is more clearance, because then the comparison with the gain of function experiments of PCSK9 would have made more sense. In here, it's a little confusing because in both cases you see a drop. When you overexpress PCSK9, you see a drop, and then when you reduce it, you see a drop. That's confusing. But I need you to understand that what they're measuring here is what's left in the brain, right? So in a way, seeing less A-beta left in the brain is a reflection of more clearance. Does that make sense? No, the effect is the same. It's just that they're displaying it differently. In vitro, they saw how much A-beta made it through the cells. Here they're measuring how much A-beta is left behind in the brain. So seeing less A-beta left behind in the brain is an indication that more A-beta made it out. So there are mirror images of the same phenomenon. In one case, we overexpress PCSK9, we see less transport. In this case, we inhibit PCSK9 and we're seeing more transport. And because there is more transport out of the brain, there is less left in the brain. Okay? And in these experiments, they didn't use regular mice. They used mice that are naturally prone to producing more A-beta. If you take an absolutely regular, healthy young mouse, they may not be producing enough A-beta for you to see, right? Because healthy mice will clear this thing, will process it, metabolize it, and so forth. So to help themselves, what they did is use these 5X FAD mice that we saw before that are genetically prone to developing AD and that are genetically prone to producing more A-beta peptides. So they're using a mouse that is naturally more full of A-beta in the brain. Say it again. A young, healthy mouse would metabolize it, would get it out of the brain, send it to the liver, and in the liver it would be destroyed. That's what I mean by metabolize.
When we inject with antibodies, you're expecting it to pass the line.
Yeah, and so when we inject with the PCSK9 blocking antibodies, the expectation is that now more A beta peptides are making it into the blood, sent to the liver, and destroyed. So if we measured in the liver, who knows what we would see? But that's why, on purpose, they're measuring how much is left in the brain. And what these experiments are saying is when you block PCSK9, you have less A beta left in the brain. And the thinking is that more made it into the blood, to the liver, and out, destroyed. Through metabolism, yeah. Oh, yeah. Well, it's not these are ELISA measurements, whereas the other one was percentages, because the other one you have to represent as a percentage of what was on the other side, right? So in this case, what they're showing you is the raw measurements of how much was present in these brains after the experimental treatments. So they're basically, these are milligrams. So these are picograms of soluble A beta per milligram of protein. So, yeah, soluble and insoluble, and the changing scale here may have to do with how much there was in each case. So they just need to adjust those things. Okay, so you block PCSK9 in a mouse by injecting it with PCSK9 blocking antibodies, and as expected, there is less A beta left in the brain. But if you do the exact same experiment in mice that, once again, are in an 5x FAD background, so they're more prone to producing a lot of A beta peptides, but you take out a copy of the LRP1 gene, so through controlled genetic crosses, you basically cross these 5x FAD mice to knockouts, so the progeny will be 5x FAD, but also miss a copy of the LRP1 gene. Now, what you see is that there is no effect of adding PCSK9 blocking antibodies. And the overall rate of A beta in the brain increases in both cases, both for the soluble and insoluble. And once again, this is a mirror image of what we're seeing before. It makes total sense that if there is less LRP1 in these mice now, there's going to be less transport of A beta across the endothelial cells, and therefore there's going to be more A beta left in the brain. So we're seeing two things at once, both of which are consistent with the model. We take out LRP1, and two things happen. First, the A beta levels in the brain increase, so we see that overall increase compared to controls. And second, we've now made these mice refractory to the effect of PCSK9 inhibition. Now they don't care if you inhibit PCSK9 because you've taken away the intermediate, and now PCSK9 doesn't do anything. Does that make sense? Yeah. Cool. So they take these mice and they use them in several other experiments. In this first experiment, they measured levels of A beta left in the brain. There is this other experiment in which they go ahead and measure the formation of plaques after a while. These mice, once again, are prone to developing plaques much sooner, younger than regular mice because they're genetically modified such that they produce these plaques way more than wild-type control. And what they see is that when they add the PCSK9 inhibitor, there is the microscopy images up there, and then they see that there is a significant decrease in formation of plaques, both in the prefrontal cortex and the hippocampus of these mice. So now we can put the two observations together, and it makes total sense. There is less A beta left in the brain when you block PCSK9. It's pretty reasonable that there would be lesser plaque formation because there is less peptide left behind to make those plaques. Right? So not only is there a decrease in the levels of A beta peptides and therefore oligos left behind, but also a reduction in plaque formation. And then they go for a series of behavioral experiments that address cognitive function. And I wanted to kind of quickly introduce what the experiment looks like. So they measure memory, and there is two types of memory, right? Broadly speaking. There is associative contextual memory. That's when you kind of remember that something happened in a given context. And then there is conditional learning, conditional memory. And that is when you associate a stimulus to something that happened either immediately after or simultaneously, right? So the experiment goes something like this. They have mice in a regular environment. Then they move them to an environment that presents some sort of distinct context. With PowerPoint, I just drew a whole bunch of dots. I don't know what the experimenters did, but they probably had stripes in the cage or some shapes on the walls, whatever, that told these mice, oh, this is a new environment. They leave the mice in that environment for about 3 minutes. And the last 30 seconds of those 3 minutes, they do two things. They start playing a tone that the mice hear, and towards the end of that 30-second tone, they shock them, right? So what happens is, the day after, they take the same mouse, place them in a regular cage, no blue dots, no stripes, no shapes on the walls. So they're like, oh, nothing bad is going to happen here. But then the experiment itself is they move those mice to the context where the tone and the shock happened the day before. And what they measure is how much those mice freeze in that environment. A regular mouse will remember, oh my God, I remember this room. Yesterday I got shocked here, right? So a regular mouse would freeze. That's an indication of that they're experiencing fear, right? They freeze. So that's the measurement, is the percent of or condition freezing, right? And what they see is, let's take a look at the control here. The control is not freezing much. It's about 25% freezing. It's like they don't remember, right? They're not freezing much. That makes total sense. These are 5X FAD mice. These are mice that are prone to developing Alzheimer's disease. They're prone to developing cognitive impairment. Little freezing is what, in this case, you would expect. These mice, because they are FX FAD, they're very forgetful, right? So they don't remember much, they don't freeze much. If you take those same mice and then you had injected them for several days before this experimental or this behavioral assay with the PCSK9 inhibitor, now they freeze more, significantly more. It's like they're remembering better. There's improved cognitive function. They're remembering better that this environment meant bad news yesterday. Rocio? Same mice, the gray bars, you didn't inject anything into them, or you injected saline. And then the the 5 times FAD mice, white bars means that you injected the PCSK9 blocking antibodies into them.
So, I mean, it's the same 5X FAD that they get out better?
These are two separate group of mice, but if these groups are large enough, this variation here means that some mice remember better, some mice remember worse, but as a group, the FAD mice didn't remember so well. If the FAD mice you had injected is a separate group of FAD mice, but they're genetically identical, so a separate group of mice that you injected or inhibited PCSK9 in, now they'll remember better. Okay, not better, because there is no significant difference between the wild type and the treated 5X FAD. But what this is showing is that the treated Alzheimer's prone mice, now remember as well as a wild type. Essentially, what this experiment is saying is you take one of these Alzheimer's prone mice, you inject them with CSK9, you've quote unquote cured them. Now they can remember just as well as a normal mouse, right? And then last but not least, the LRP1 knockout that experienced a lot of A-beta accumulation in the brain, perhaps not surprisingly, they're pretty bad at remembering that context. So they don't freeze at all. Even though they got shocked in that context yesterday, today they don't remember. And if that sounds like the kind of amnesia that some Alzheimer's patients experience, and particularly later on in the progression of the disease, that's exactly what it is. These LRP minus mice or heterozygote mutants accumulated a lot of A beta, they will form a lot of A beta plaques that will be toxic to the neurons, and now these mice don't remember that in this environment they got shocked yesterday. So they don't freeze. We're going to go through an important control now, but I wanted to turn it around and make it into a question. So we're saying here that not freezing is a reflection of they've lost memory, right? So why else, other than memory loss, would a mouse not freeze in a context that the day before was paired to a shock? If you were devil's advocate and you see these mice in these peculiar context not freezing, what would you propose might be going on? If it's too hot or too cold, but we need to assume that the temperature was the same in both cases, yeah. All right, so that would show up in the controls as well, right? So if the mice have not been trained enough, so they haven't really learned the association between the environment and the shock, then you would expect that the wild types wouldn't freeze either. But I love that point. So elaborate on that, because the control that I want to show next is related to that, right? So these mice that the FAD mice and these LRP knockouts are freezing less. And we say, oh, they forgot. What's an alternative interpretation? That's right. So maybe the problem isn't so much with remembering the connection between the context and the shock. They never really learned it in the first place, right? So see how it's very along the lines of what you were proposing. So maybe the problem isn't about recalling the connection between context and shock. It's about establishing the connection between context and shock in the first place. So how did the authors control for that? With this experiment. They took the mice, kept them in a non-contextual environment, a regular cage, and then they played the sound. And the sound was intimately associated with the shock. And guess what? Now, all of the mice, no matter what the genetic background is, whether they have LRP1 or not, whether you block PCSK9 or not, all of them freeze to about the same extent. And that's an indication that all of them had learned that with the sound comes the shock, right? And it's an indication that they did feel the shock, they did not like it, right? And then the day after, when they hear the sound, they freeze. But then some of those mice, if you put them in an environment that was loosely associated with the shock, some of them remember, the wild types and the Alzheimer's mice with the CSK9 treatment, but then the others don't. Did that make sense? All right, and in the last couple of minutes, I wanted to address this. So far they've shown us that PCSK9 overexpression, when they put a lot of PCSK9 in the endothelial cells, lowers LRP1 expression. They also showed us that overexpressing PCSK9 lowers A beta transport in an LRP1 dependent manner. And then they showed us that the opposite, blocking PCSK9, will lead to increased transport, more clearance of the A beta, getting it out of the brain and improving their memory and cognitive function and whatnot. So, if you have to put it all together in a model that explains why injecting these PCSK9 blocking antibodies helps the mice with memory and cognitive function, how would you do it? What would be the model? If you had to explain to someone who doesn't know about this paper how this works, how would you do that? Ooh, liver to metabolize. Okay, so liver to metabolize. But you need to get them out of the brain first, right? So you want A beta out. That is normally done by what? LRP1, right? And what is CSK9 normally doing to LRP1? degrades LRP1. So what CSK9 is really doing is inhibit LRP1. And this LRP1, where is this LRP1? You're saying everywhere? Cerebral. Well, the endothelial cells, right? So this LRP1 is the one that is sitting in the endothelial cells that moves A beta from the cerebral spinal fluid to the blood, right? So this would be the endothelial LRP1. Correct. So this would be the model. But guess what? They went ahead and measured LRP1 expression in these mice that were injected with PCSK9 antibody in the brain. So they took the brain, they depleted those brain fractions from capillaries. You can do that by basically dissociating all of the cells in the brain through enzymatic means, and then you use experimental laboratory tricks to get rid of the endothelial cells, like move them aside, and then everything else that you have left is brain, neurons and glia, astrocytes, microglia, and all of that stuff. And when they did that, they saw no change in LRP1 levels. But then when they took the same mice and took liver fractions, they saw a mild increase in the levels of LRP1 in the liver. So what the authors are saying is that when even though manipulating CSK9 activity may have an effect on brain endothelial LRP1, what they're saying is that in these particular experiments, what they're thinking is that manipulating CSK9 altered the levels of LRP1 in the liver. And it was through this sync model that this whole thing works, right? So in other words, do I have the, no, that was the last slide. So what they're proposing is I inject PCSK9 blocking antibodies. I don't do much to LRP1 in the brain, but I do increase LRP1 in the liver. So even if LRP1 in the brain is not changing and the same amount of A beta is being pumped out of the brain, it's going to get much more efficiently clear by the liver. So a lot more A beta is destroyed by the liver, a lot less A beta makes it back to the brain, and in the long run, what that does is it reduces the levels of A beta, which they saw, and improves cognitive function, which they saw. Does that make sense? For those of you who didn't get a chance to participate today, the question that I asked in Canvas has to do with the fact that they're looking at brain cells like neurons and astrocytes and microglia, but they're not really looking at those brain endothelial cells. So how would you propose to look at that instead? Okay?